Hey, today I'm going to talk about a project that has been financed by the HIF grant, that is leading reach-wide lab. And we're going to present a new F class of antibodies, that is the F9 antibodies that are capable of neutralize autologous tier 2 viruses in NHP that were immunized with the liposome conjugated well-ordered trimers. I'm going to describe first the trimers that we use for this work. We use the NFL TD-CC 16055 trimers that are derived from the clade C strain. These trimers, they have a native flexible linker that holds together the GP120 and the GP41, and they are replacing the furin. That's why those trimers are cleavage independent. They also contain the IP mutation, 8 trimer-derived residues that were reported by Javier Guenaga in 2015 that conferred trimer stability, and the disulfide bond that was also described by Javier in 2015, where this disulfide bond holds the trimer in the perfusion state. And they also contain the N-glycan 332 inserted. So these trimers were displayed in the liposome. These liposomes were described last year in 2016 by Richard White lab. And after the display of these trimers in the liposome, we wanted to check the stability of the trimer conjugated liposomes. We checked the stability at 4 degrees in PBS, at 37 degrees in PBS, and at 37 degrees in matrix M. That was the adjuvant that we used for the immunization studies. We also confirmed that these trimers maintain a well-ordered conformation by testing the binding towards antibodies, broad neutralizing antibodies that are trimer-specific and antibodies that are CD4 binding site. And also we saw the lack of binding towards non-broad neutralizing antibodies. So with these liposomes displaying the 16055 NFL trimers, we wanted to analyze the quality of the vaccine-induced antibody responses in NHP. And for that, we had 2 groups of animals, the soluble group, in which we had 6 animals, and the liposome group that we have also 6 animals. All those animals received the same amount of trimer, 100 micrograms, matrix M, 75 micrograms. So we started to analyze the immune response, and then we started to analyze the early immune response after the first and the second immunization. And when we analyzed the titers of IgG binding antibodies against ENF, we saw that the levels were really similar between the 2 groups. We didn't see any difference. And then we took lymph node samples from the animals, and we analyzed these lymph node samples by immunofluorescence. This work was done by Gustav Lindgren, and we stained the cells with CD3, PD1, and K67. And as you can see in this figure, you can see per lymph node all the germinal centers that are characterized by being a conglomeration of cells that express PD1 and K67 that are surrounded by CD3. So when you take one of those germinal centers here in the red square, you can manually select the germinal center, and using a cell profiler software, you can quantify the fluorescence signal inside of that germinal center. So by doing that, we were able to calculate the individual GC areas in the liposome and in the soluble and the liposome group, and also the number of stir phage cells containing inside the GCs. So we saw that the liposome group has larger GCs compared to the pre-samples, but most importantly has larger GCs compared with the post samples in the liposome group. And when we analyzed the number of the FH cells contained in the germinal center, we saw also that the liposome group was having an increase of the number of the PD-1 positive cells compared to the pre-samples, and most importantly against the soluble post-immunization samples. So with this, we conclude that the liposome has an effect on the early immune response in the GCs. Then we analyze the kinetics of the whole response during the whole regimen. We have 5 immunization regimens, and then we check for the antibody titers against ENF in both groups, in the soluble group and the liposome group. And we saw that there was no difference in the level of IgG titers between those two groups, and that we reached a peak of antibody response after the second immunization that was not further boosted with any of the immunizations. With this in mind, then we analyze also the neutralization activity against the clade C 16055 isolate, and we saw that the liposome group, as you can see here, has a preferential achieving the neutralization autologous activity compared with the soluble trimer group, or where only one animal has autologous neutralization activity. In the liposome group, the autologous neutralization activity was consistent, and some animals have higher neutralization activity than others. Then we analyze the statistics against the soluble and the liposome group after the third, fourth, and fifth immunization, and we saw that the levels of the neutralization activity were statistically higher for the autologous neutralization after the fourth and fifth immunization. And in comparison, when we analyze the tier 1 viruses, like SF162, we didn't see any difference in the neutralization activity between the soluble and the liposome groups in any of the time points analyzed. So we wanted to analyze the specificities that were driving the neutralization activity against the autologous virus. And for that, we took one of the animals with the highest neutralization activity, the 11 animal. And from that animal, we isolate ENF-specific memory B cells. So we did consistent standardization of the panel at Karolinska using protocols that were described previously by us in the group. And with this panel, we effectively sort single ENF memory B cells from this D11 animal 2 weeks after the third immunization. And we were able to isolate 18 monoclonal antibodies from one single cell-sorted plate. Those antibodies, they have the characteristic that all of them bind the ENF trimer, and some of them, they are not able to bind the GP120 monomer, which indicates that they are directed against the GP41, and some of them, they bind the V3 peptide, indicating that they are directed against the V3 region. And when we analyze the neutralization activity, we saw that some of the antibodies, they have autologous tier 2 neutralization activity. We further analyzed the genetic of all of these antibodies, and we analyzed the CDR3 length of the heavy chain. We saw that there is between the 8 and 21 amino acids, and that the autologous tier 2 neutralizing antibodies have the same length, 13 amino acids. And in terms of the somatic hypermutation, at the amino acid level, we saw that the mutations were between 2 and 80%. That is consistent with what we have been previously reported. And the autologous neutralizing antibodies, they will have a somatic hypermutation between 8 and 30% at the amino acid level. So we were very interested in trying to characterize which are the epitopes that these antibodies are binding, and as soon as we finish with the analysis of the antibodies and the isolation of the antibodies in the lab, we send them to Ridge-Wide lab to further characterization. Here in this lab, we do epitope mapping, and this epitope mapping started with the cross-competition binding assays. So we did cross-competition against first the autologous tier 1 antibodies, and then we saw a strong cross-competition between them. When we did the cross-competition against B1, B2 broad neutralizing antibodies, we saw weak cross-competition. And when we do cross-competition against B3 glycan and OD glycan, we saw intermediate cross-competition. When we analyzed the CD4 binding site cross-competition, we saw weak and intermediate cross-competition. And to further analyze this, we analyzed the binding of the antibodies to the GP120 D368R deleted mutant that has a defect in the CD4 binding site. And then we also didn't see any binding to this mutant, which means that these antibodies are not CD4 binding site directed. And when we finally analyzed the GP120 GP41 the cross-competition, we didn't see any cross-competition, meaning that the antibodies are not interacting with these regions. So in order to better define the regions that the antibodies are recognizing, we did target variable deletions, and then we analyzed the binding of the antibodies by ELISA. So we did first deletion of the B3 region, and then the antibodies were still able to bind. Then we did the deletion of the B1, B2 region, and then we abrogate the binding. And then we did the deletion of the B1 and B2, and then we saw that the abrogation of the binding was present in the deleted B2 variant. And then to further characterize this, we did the deletion of the B2 loops, B2.1 and B2b, and then we saw abrogation of the binding in the B2b, meaning that this is the region towards which the antibodies are having the main contacts. To further define the motifs that the antibodies are binding, we did B2 loop alanine scan. For that, we need to construct a series of B2b alanine mutants to do neutralization assays. And as we can see in this table, when we replace the amino acids in the position 185 to 186, we saw loss of the neutralization activity, meaning that these amino acids are important and are an important part of the motif that the antibodies are recognizing. But further alanine scans, mutants are needed to completely define the motifs that the antibodies are binding in the B2B loop. So that work is in progress. So because we have in mind that now that the antibodies are binding the B2 region, so we wanted to check if the deletion of the B2, B1 can abrogate the binding of the antibodies. And for that, we need to create first a B1, B2 deleted trimutant. And this probe was created, and we analyzed that the deletion didn't affect the recognition by conformationally sensitive antibodies, and that actually we abrogate the binding of B2 specific broad neutralizing antibodies has PG9. So with this delta B2, B1 trimute, we did absorptions of the monoclonals and the plasma of the animal that we isolate the antibodies, and we saw that the wild type absorbed the autologous plasma neutralization, while the deleted B1, B2 did not. And we have as a control an antibody that binds the B2 region, PG9. So we see the same trend, and we have as another control an antibody that binds the CD4 binding site, in which we didn't see the same trend. So we further analyzed the activity in the plasma of other animals, and we saw that in all the animals that were having autologous neutralization activity, the wild type is capable of absorbing the autologous plasma neutralization, meaning that they are directed against B1, B2 antibodies. And with this, we conclude the F9 class antibodies can recapitulate the serum autologous neutralization activity. In order to get a low-resolution picture of how these antibodies are interacting with the trimer, we wanted to do a negative staining electron microscopy, and for that we need the antibody that has the highest affinity. So we measured the affinity of those antibodies by a core, and we found that the Fab of the F9 antibody has the highest affinity. So we used that antibody to do the negative staining electron microscopy. And here we can see a compilation of the pictures that were taken of the trimer making a complex with the Fab of the F9 antibody. From these complexes, we saw that 55% of these complexes have the trimer with 3 Fabs, as you can see here in this picture. 33% has only the interaction with 2 Fabs, 6% with the interaction with 1 Fab, and 6% of the trimers, they don't have the interaction with any Fab. So by taking the complexes that were having interaction with the 3 Fabs, we recreate a 3D reconstruction density. And with this density reconstruction, we were capable of fitting the VG505 SOCIP664 trimer in complex with the F9 Fab using different programs. And we can see that the antibodies here in red, they interact with the B1 and B2 region, and this is consistent with what we saw in the alanine scan and in the punctual deletions, when we saw that the B2B loop is the main interacting motif of the antibodies. And then when we check the side view of this model, we saw that the antibodies here in red, they approach the B2 by a lateral angle. And further, when we did docking also using the BRCO1 antibody, we saw that the F9 antibodies are on top of the BRC antibodies, and that correlates with what we saw in the alanine scan, where we saw cross-competition with the CD4 binding site antibodies, including the BRCO1. When we compare the binding of the F9 antibodies and the PG5, we see that the F9 antibodies, they have a different approach, and the PG9 antibodies, as we already know, has a more vertical approach toward the apex. And that also correlates with what we saw in the alanine scan, when we saw weak cross-competition with B1, B2 broad neutralizing antibodies. And finally, we analyze the composition of the glycans that are surrounding the V2 regions that are the target of these antibodies. And we see that the antibodies, the F9 class antibodies, they penetrate the glycan shield in order to interact with the V2 region. So in summary, we found that the 16055 autologous neutralization in NHP immunized with the 16055 NFL TDCC trimers is mediated predominantly by targeting the V2 region, that the liposome array, the platform, induces superior GC responses and neutralization activity against the tier 2 autologous viruses, that the isolated monoclonal antibodies with autologous neutralization recapitulate the plasma neutralization activity, and that the F9 class monoclonal antibodies target the V2 by using a lateral approach toward the apex. Thank you very much for your attention.